Okay, all right, so good afternoon, everyone. So just a reminder that we are now in the midst of our term quiz 2 rebuttal phase. So if you're planning to submit a rebuttal to any one of the questions on term quiz 2, has to come in by Friday at the end of the day, and then I will try to get to them at some point. Don't know when that will happen necessarily with the Thanksgiving weekend ahead of us, but somewhere along the line I'll fit that in between bites of turkey. Other than that, yeah, I don't know. Any questions about things before we continue on with the lecture here? Everybody's doing okay? Yeah? Okay. Okay, so we are going to continue on here with our electrophysiology lecture. We got introduced on Tuesday to the resting membrane potential. And just recapping a few of the things we talked about, we defined the resting potential as this charge difference that exists across the cell membrane, positive on the outside, negative on the inside. We noted that all cells have a resting membrane potential, and that's because it can be used as an energy source to power some kind of work or biosynthesis within the cell. And we, in fact, looked at an example of that. We looked at the active uptake of glucose into cells by the sodium glucose cotransporter. And then we ended kind of noting that because the plasma membrane that separates the extracellular and intracellular fluids is so thin, that ultimately we don't actually have to have that many absolute number of ions moving across the membrane to create these membrane potential values. And so that kind of, again, jived a little bit with our conversation of the law of macroscopic electroneutrality, where we said, like, ion solutions should generally be electrically neutral, approximately. And so, yeah, we might have, a small number of ions different. We might have a few more positive charges on the outside of the cell and a few more negative charges on the inside of the cell. We're not quite, ion for ion electrically neutral. But it only has to be approximate, all right? So that's kind of where we were at the end of the lecture on Tuesday. Where I want to move on to this afternoon is to really get into more of the nitty-gritty details here of exactly how the resting membrane potential actually forms. We kind of got a bit of a taste of that on Tuesday as well. We basically said that you're going to have these different concentrations of ions in the extracellular and intracellular fluid, and then you're going to have ions kind of moving across the membrane down their concentration gradients, and because these ions are charged, that's going to lead to a charge separation, a charge difference across the membrane. But I want to get into the details of that a bit more. So this figure that I have here is showing us the ion composition of the extracellular and intracellular body fluids. And what is abundantly clear when you look at this figure is that the extracellular fluids and the intracellular fluids have very different ion compositions, right? You can see that the extracellular fluids are very rich in sodium and chloride. They also have more calcium and bicarbonate than what we see in the intracellular fluids. And then when you look at the intracellular fluids, they are particularly rich in potassium and phosphate. They also have more protein than the extracellular fluid, and also typically more magnesium. So quite a bit of a difference here in terms of the ion composition of these two fluids. And as I've already said, it's this difference in ion composition that largely gives rise to the resting membrane potential, and we're going to talk about how that occurs in just a moment. But I want to address another question here first, which is just like, well, why are the intracellular and extracellular fluids so different in terms of their ion composition, right? We sometimes sort of gloss over that point and take for granted that they are, but why are they? And so the first answer to that question is that there are some ions that are just membrane impermeable. So they haven't got the ability to just move across the membrane and kind of equilibrate their concentrations on both sides of the plasma membrane. And the two ions I've highlighted here as being membrane impermeable are phosphates and proteins. We'll start with proteins first. So the first thing to appreciate here is why are we even including proteins as an ion on this diagram? Well, it's because most proteins have a net negative charge under physiological pH conditions. And so they're included here on our figure because they are in fact anionic under physiological pH conditions. And why can't they cross the membrane? Well, because they're just too damn big, right? They're too big to just go across the plasma membrane. And so we have this abundance of predominantly negatively charged proteins that are effectively trapped inside the cell because they're too large to get across the membrane. With respect to phosphates, so why can't they get across the membrane? Well, largely they can't get across the membrane because there are no transporters to allow them to cross the membrane. We're going to talk more about channels to allow ions to move across membranes in a little bit here, but there are no transporters, there are no channels that allow for phosphate to pass across the membrane. And also, anything that's phosphorylated is also not able to cross the cell membrane, right? So it's not just phosphates that can't cross the cell membrane, it's also things that are phosphorylated. And I just want to dwell on that point for just a little bit here, because we actually take advantage of this, or the cell takes advantage of this during metabolism. If you think about the first step of the glycolytic pathway, where you take glucose and you use ATP to phosphorylate it, right? So this is the hexokinase reaction. I don't know if you've ever really stopped and thought about, like, why is the cell doing this, right? We think of glycolysis as a pathway that's supposed to be yielding and producing ATP, but right in the very first step, it's consuming the very resource it's supposed to be making. Why is it doing this? Well, the answer is because glucose, as we learned last week, there are glucose channels. We talked about the GLUT1 channel, for example, that can let glucose get across cell membranes. Glucose can generally get freely across cell membranes because there are channels for it. But once the glucose comes inside the cell, if you phosphorylate it, now it's trapped inside because it can't get out through that glucose channel. It's not glucose anymore, it's a phosphorylated glucose, and there are no channels for phosphorylated compounds to pass through the membrane. So if you actually were to look at the entire glycolytic pathway, you'll find that every intermediate in that pathway is phosphorylated. That's to trap it in the cell. That's to make sure it can't get out. You don't want to be halfway through glycolysis and like, oh fuck, all my intermediates are leaking out of the cell and basically I just, aren't getting any energy out of this now. You have this mechanism through phosphorylation to trap things in the cell, and that's why the membrane has evolved to be impermeable to phosphates and phosphorylated compounds. But what about the rest of these ions? These other ions are all membrane permeable. There are channels that allow them to move across the membrane. So, why don't we just equilibrate? Why don't they just equilibrate, right? Okay, we know the phosphate is trapped, we know the proteins are trapped inside, but why don't all these other ions equilibrate across the plasma membrane if they have channels that would allow them to move across the membrane? Well, what I've done here now is I have equilibrated all these other permeable ions, and so let's figure out what the problem with this would be. The problem with this would be that if you added up the total concentration of cations and anions now in both your extracellular and intracellular fluids, you come to see, if you look at the math here, that you've got obvious charge imbalances now in your two fluids, right? The intracellular fluid at this present moment is too negatively charged, right? And the extracellular fluid is too positively charged, largely because you've got a bunch of negatively charged things that are trapped in the cell and can't equilibrate. So if all the other ions that were permeable just equilibrated their concentrations across the membrane, we would have a significant violation of the law of macroscopic electroneutrality, right? We said we can tolerate a little violation, approximately, but here we're talking millimolar concentration differences. That's not a negligible, approximately violation there. That's like a big, huge violation. So we have to recognize that the other ions can't just equilibrate their concentrations across the membrane because it would cause this huge charge imbalance, this violation of the law of macroscopic electroneutrality. So what should happen to kind of bring us back to charge balance is we might just expect that the positive charged ions would kind of move into the inside of the cell, kind of drawn by its current negative net charge, and the negative ions would kind of move out of the cell, right? Drawn out there by the net positive charge. So kind of looking from here, we had this equal distribution of those ions that are permeable, and we said now we're envisioning that the positive ones will drift in and the negative ones will drift out. And now you can see this gives us charge balance, right? We've now obeyed the law of macroscopic electroneutrality just by that movement of positive ions into the cell to balance out the negative ones trapped inside, and the movement of negative ions outside the cell to balance off the excess positive charge. This movement of ions to try and establish charge balance across the, or charge balance within the fluids on either side of the membrane, that shift of those ions is called a Donan equilibrium. So anytime you have ions kind of shifting across a membrane to try and make sure that you have the fluids on both sides being, electrically neutral, that's referred to as a Donan equilibrium. So that's what we just saw there. So we've solved one problem. The law of macroscopic electroneutrality is now obeyed, right? We have equal concentrations of cations and anions in both our intracellular and extracellular fluid, but now another problem has arisen. If you add up the total ion concentrations in both these fluids, they are now very different. We have, much more ion content in the intracellular fluid now than we do in the extracellular fluid. And so yes, the law of macroscopic electroneutrality is obeyed, but now we've created a significant osmotic gradient. And now water is going to be wanting to come into the cell to try and balance that osmotic gradient out, and that's not a good thing, right? We don't want water flooding into our cells, causing our cells to swell and potentially burst. So we solved one problem, but in doing so, we created another. So now we have to solve another problem. And the way that the cell solves this problem is through the expression of the sodium-potassium ATPase, right? This is another enzyme that is ubiquitous, right? We see this expressed in all cells within the human body. And so you're probably familiar with the sodium-potassium ATPase, but as is pictured here, it's obviously using the energy of ATP to power its function. And then what it's doing is it's taking 3 sodium ions from inside the cell, and it's pumping those ions into the extracellular environment. And then it's picking up 2 potassiums from the extracellular environment, and it's pumping those into the interior of the cell. So what we have to appreciate here is that we're pumping more ions out of the cell than we are pumping into the cell. We've got 1 net ion being pumped out with every ATP that we're using. And so that's good because we already had too many ions in the cell and not enough ions outside the cell. So by pumping ions out of the cell, 1 net ion per cycle here, we're helping to restore this osmotic balance. And so here I'm going to shift my sodiums and potassiums, right? Lots of sodium is going to move out of the cell now, and potassium is going to come into the cell, and that's all because of the sodium-potassium ATPase. On top of that, most cells have a calcium ATPase that just takes calcium from inside the cell and pumps it to the outside of the cell. And so that's good again, because that's going to help us to get more ions out of the cell, help us to achieve this osmotic balance. You can imagine now that because of the sodium-potassium ATPase, not only is it pumping 1 net ion out of the cell, but it's also pumping 1 net positive charge out of the cell. And the calcium ATPase is also pumping, 2 positive charges out with every calcium. Now you're thinking like, oh fuck, we're back to this whole electrical imbalance problem. Well, we'll solve that very simply with a second Donan equilibrium, right? These negative chlorides and negative bicarbonates will just kind of shift out. They'll follow those positive charges we've pumped out. So we have this second Donan equilibrium. You'll sometimes hear people talk about the double Donan equilibrium in the context of resting potential. It's these 2 Donan equilibriums that we just saw kind of working together. But now, because of these 2 Donan equilibriums, because of the action of the sodium-potassium ATPase and the calcium ATPases, we've actually now got to a point where we have both electrical neutrality in both the intracellular and extracellular fluids, and now we have approximately the same ion concentrations in the intracellular and extracellular fluids, so we don't have an osmotic gradient. And what you notice is the end result of this whole process is we're back to where we kind of started in this conversation. We're back to seeing why the extracellular environment is very sodium and chloride rich. and also rich in calcium and bicarbonates? And why is the intracellular fluid rich in potassiums and phosphates and proteins? We can see how that's arisen through these two donor equilibria and the action of the calcium ATPases and the sodium potassium ATPases. Now you might also say, well, we aren't quite at perfect osmotic balance here, and that's because, of course, we're only thinking about ions in this particular figure, but of course there are other things that are dissolved, especially if we think about the extracellular fluids. I mean, you're going to have things like glucose, right? I mean, the human body has about 5 millimolar glucose there. I put urea as another example, but there are other solutes here, aside from just ions, that also contribute to the osmolarity, generally to a lesser extent, but they would make up any kind of imbalance here that we have remaining. So the idea is that we, again, through the action of these two donor equilibria, through the action of the sodium potassium ATPase and the calcium ATPases, we have managed to obey the law of macroscopic electroneutrality. We have managed to maintain a perfect osmotic balance between the extracellular and intracellular fluids, and the end result of that is these different concentrations of ions that prevail in the intracellular and extracellular fluids. One thing I want to kind of help you appreciate here is that we spend a lot of energy doing all of this. You know, the sodium potassium ATPase alone accounts for about 30% of our BMR. So that's a lot of energy being spent just to kind of, prevent these osmotic gradients and prevent these. I mean, you don't really have to spend energy to prevent the electrical imbalances. They'll just kind of settle themselves. But to prevent the osmotic imbalance, and that's where the sodium potassium ATPase came in, like we're spending a ton of energy to do this. Right? And then that's not even accounting for the calcium ATPases, which probably add another 5 or 10%. So under basal metabolic rate conditions, we're spending a lot of energy on just trying to prevent our cells from flooding with water, right? But obviously that's a worthwhile investment, because if all your cells just swelled up and bursted, I mean, the conversation of how much energy does it cost to not do this would be moot, right? It's like you saying, like, well, I don't really want to buy that sub pump to stop my basement from flooding, but of course if the basement floods and it's just like a moldy old house, well, it's kind of a moot point how much it costs to have that sub pump to get the water out, because I can't live in a mold infested house. Like, it's a necessary energy cost is kind of the point I'm making here. The other point that I want to drive home here at this point, because this is a very common kind of misconception about resting membrane potential, obviously the sodium potassium ATPase, as we already said, is an electrogenic pump, right? Meaning it's pumping one net positive charge out of the cell with every cycle it goes through. And the calcium ATPase is the same way. It's pumping out 2 positive charges, right? This 2 plus calcium with every cycle that it goes through. And so where some of the misconceptions come in is oftentimes people will think, oh, well, the resting membrane potential is being generated by the action of these pumps, right? That they're pumping positive charges out of the cell, and that's making the exterior of the cell positive and the interior of the cell negative. The reality is that those 2 electrogenic pumps, their direct contribution to the resting potential is very small. They are certainly not the primary generator of the resting membrane potential. Their job is to maintain the sodium, potassium, and calcium ion concentration gradients that we saw on the previous slide. And that, as we'll see in a moment, it's those gradients that drive the formation of the resting potential. These pumps do not directly contribute much to the resting membrane potential. And I have a little bit of data to illustrate this point. I wish I had better data, but this is not something that people necessarily publish great figures about. And hence you can see this figure looks like it came straight out of the 1950s off some guy's typewriter. But this was a study where researchers were looking at what happens to resting membrane potential if we inhibit the sodium potassium ATPase. Now before I show you what happens here, one point I want to draw your attention to here. Remember how yesterday or Tuesday we had discussed how we always measure the resting potential by putting our recording electrode inside and our reference electrode outside, right? So we always measure resting potential the same way. And pretty much every known cell has a negative resting membrane potential, because we're looking at what's inside where it's more negative compared to what's outside where it's more positive. Because people assume that you know that as physiologists, you'll often notice that they don't actually include the negative signs on figures. You know, when you look at that figure, you don't see any negatives in front of the 100 or the 90 or the 80 or whatever. And it's not clear to me whether this is meant to be like negative millivolts or just a dash between the name and the unit. But there's very often where you will see that researchers just ignore the negative sign. And they recognize that, like, hey, you're an intelligent physiologist. You know resting potential is negative. I don't have to put it up there on the figure. You know it to be the case. I want to emphasize that because sometimes you might come across this in some of the figures that maybe I pulled from, you know, some of the papers for the term quiz questions or something. I don't want you sitting there thinking like, oh, there's positive resting potential values? That's kind of weird. If there were positive values, I would definitely spell that out very clearly so you would know. But if you just see like a figure like this and you don't see negative numbers, assume they are negative. And assume that the person just didn't put negatives in there because they thought they were dealing with intelligent people who know physiology and know that those values are negative, okay? All right. Now, if we look at the actual data here, so they're measuring resting membrane potential over time, and right here, they add this compound called wabane, which is an inhibitor of the sodium-potassium ATPase. You can see actually in the diagram here, they sort of show where it binds. It kind of binds to the same general location where potassium binds. So wabane is an inhibitor of the sodium-potassium ATPase. If the sodium-potassium ATPase were directly responsible for contributing or directly responsible for generating the resting membrane potential, then you would expect that if you knocked it out with wabane, that the resting potential would just immediately drop down to 0, right? Because now you would have shut off the pump that was generating the resting membrane potential. But you can see here that doesn't happen, right? In these developing rat myocytes that they were using here for this study, at best we saw the membrane potential drop from -90 down to -60. So it looks like it was making maybe a 33% contribution here to resting membrane potential. That's actually a pretty big contribution. If you were to do these studies in neurons, if you did this exact study in neurons, you'd probably see only a few millivolt change when you add wabane, but I couldn't find any good figures and studies where they actually had plotted out their neuronal data. But the point overall here is, again, the electrogenic nature of these sodium-potassium ATPases and the calcium ATPases, they're not contributing in any significant way to the resting potential. This 33% contribution we're seeing here in developing rat myocytes is like on the extreme end of the contribution scale. In most typical neurons, again, it's maybe a few millivolts at best. What you do notice happening here is that over time, you start to see this kind of slow drift of the resting potential as it kind of slowly drifts back towards 0. And the reason that it's slowly drifting is because once you shut off the sodium-potassium ATPase, then ultimately, as sodiums and potassiums leak across the membrane and you can't pump them back, the concentration difference for sodium and potassium across the membrane starts to get progressively smaller and smaller and smaller and smaller until eventually it would just not be there. You'd eventually get to an equilibrium point where the sodium and potassium would be equally distributed across the membrane, and then at that point, your resting membrane potential would be 0. So again, the sodium-potassium ATPase is not directly contributing much to the resting membrane potential value. It's largely responsible for just maintaining the sodium and potassium ion concentrations of the intracellular and extracellular fluids, and it's those concentration differences, as we're going to see in a second, that really drive the resting membrane potential formation. So yeah, that's what I want to talk about next. I want to kind of talk next about how having different concentrations of ions in the intracellular versus extracellular fluid, like how does that actually lead to the development of a resting membrane potential? And so we're going to simplify the situation down here and just think about one ion at a time, right, just to kind of make it easy to grasp here. And we're going to start with potassium first, okay? We've already established that potassium is much more abundant in the intracellular fluid than it is in the extracellular fluid, as you can see here on the figure. And we know that, again, that's directly because of the action of the sodium-potassium ATPase pumping potassium into the cells. So what we clearly have here then for potassium is a concentration gradient. A concentration gradient that is favoring the movement of potassium out of the cell, right? Potassium wants to diffuse out of the cell down its concentration gradient. But as potassium leaves the cell following its concentration gradient, Because potassium is positively charged, as it leaves the cell, it's going to make the extracellular fluids more positive, and it's going to make the intracellular fluids more negative as it leaves. And this then is going to create an electrical gradient, right? Positive on the outside of the cell, negative on the inside. That electrical gradient is going to start pulling potassium back in, right? As it gets more negative on the inside of the cell, because potassium is leaving, that negative charge inside the cell is going to start pulling that positively charged potassium back into the cell. And so potassium is being acted upon by these two opposing gradients. A concentration gradient that is pushing it out, and an emerging electrical gradient that's pulling it back in. And one very simple question we can ask ourselves is, well, at what point will these two gradients be equal, such that there's no longer a further movement of potassium across that cell membrane? And the answer to that question is called the equilibrium potential. As it says here, the equilibrium potential is the value of membrane potential. So how much charge separation across the membrane do we have to have a net flux of the ion across the membrane be zero, because your concentration and electrical gradients are perfectly balanced. So that's the equilibrium potential. It's the value of charge separation, the membrane potential value that's necessary for the concentration gradient that's acting on this ion to be exactly balanced by the electrical gradient that's acting on this ion. And once these two gradients are perfectly balanced, there'll be no more net movement of this ion across the membrane. It will have achieved equilibrium. It'll be leaking out as fast as it's drawn back in. Now, we can actually calculate the equilibrium potential value. I feel a little bit like the screen here kind of falling apart all of a sudden. We can actually calculate the equilibrium potential value for an ion using something called the Nernst equation. And I'm going to walk you through the Nernst equation here, but I'll start with this just little like asterisk, I guess. I'm not going to ask you to do calculations, right? I'm not interested in your ability to work a calculator, right? I want you to appreciate the Nernst equation conceptually, right? Just appreciate what information it's telling us and what we can use it to do, but we don't have to worry about, you know, getting out the old calculator and doing the math. So here is the Nernst equation. We use the Nernst equation to calculate the equilibrium potential for a given ion. So we're going to do it for potassium. So you can see on the left-hand side of the equation, we only have one term there. It's the equilibrium potential. On the right side, the first four terms we have there, so one is the gas constant, R. One is temperature, so this would be body temperature. And so this is measured in Kelvin, right, so the absolute temperature. We've got another constant, which is Faraday constant, and then also the charge on the ion that we're interested in. So in this case, it's potassium, so the charge, of course, is +1. So we have those parts of the equation, and then the other part of the equation, in parentheses, we have the concentration of our ion both on the outside of the cell in the extracellular fluids, and also inside the cell in the intracellular fluids. And you can see then that we're going to apply this natural logarithm to that ratio there. So that's our Nernst equation. So as long as we have all this information, we can put it into our calculator. We can figure out what is the equilibrium potential for potassium. So that's what I've done here, right? So for the gas constant and for Faraday's constant, you know, I just said, you know, hey Google, you know, what are these constants? Now you have to be a little bit careful doing that because the units, there's lots of different units that people use for these constants, and so you need to make sure you're kind of using an appropriate unit, right, that's going to fit with the other units you're using in the equation. So if you ever do calculations and you're like, oh fuck, I got a resting potential of -1 million, you know, hopefully you'll say to yourself, that doesn't seem right, and maybe trace it back to one of these constants being in the wrong, wrong unit. I've got temperature here. So 310 Kelvin, which is 37 degrees Celsius, so typical human body temperature. And then my charge on potassium is +1. And then for the concentrations, I just kind of eyeballed it from the figure here. You know, looks about 5 millimolar in the extracellular fluid. Looks about 140 millimolar here in the intracellular fluid. So I use those values, put it through my calculator, and got this value, -89 millivolts. That's my equilibrium potential for potassium. So what is the significance of this value? Well, it tells me that given this concentration gradient, right? 5 millimolar outside, 140 millimolar inside. Given that concentration gradient, potassium will leak out of this cell until a membrane potential of negative 89 millivolts develops. And then at that point, that electrical gradient, that negative interior of the cell, will be pulling the potassium back in as fast as the potassium is leaking out down its concentration gradient. We'll have these two gradients equal, will be at an equilibrium, and we call the membrane potential at this point the equilibrium potential. The next thing to appreciate is that if at any given time the membrane potential is not equal to an ion's equilibrium potential, then that means that that ion is not at equilibrium, and it's going to be moving across the membrane in whatever way it needs to try and get to its equilibrium potential, right? Everything wants to go to equilibrium. So if you have an ion and it's got an equilibrium potential of negative 89, but the membrane potential is not currently negative 89, that ion is not at equilibrium. And so it's going to move in whatever way gets it to equilibrium. So let's imagine for a moment that the membrane potential right now were negative 60. Okay, clearly negative 60 is not equal to negative 89. So potassium is not at equilibrium. Which way is potassium going to have to move to get to equilibrium? Which way is it going to have to go? I'll throw this out there as a question. Which way am I going to have to go? If I'm potassium, it's currently negative 60. I want to get to negative 89. Which way do I have to move? I have to keep moving out, right? Currently, the membrane potential is not negative enough to be at equilibrium for me. I have to keep moving out to take more positive charge out to make the membrane potential more negative. I want to get to that negative 89. And of course, by contrast, if the membrane potential were currently negative 120, right? Again, potassium is not at equilibrium. In that case, potassium would have to come in, right? In order to bring positive charge into the cell to get it to that negative 89 equilibrium. Okay, so that's an important thing for us to keep in mind, that ions are always going to move in whatever direction is necessary for them to try and get to their equilibrium potential. Okay. I had a little note here about, doesn't this movement of potassium violate electron neutrality? We already addressed that in the lecture on Tuesday. Yes, it does, but again, there aren't that many ions that actually have to move to create this membrane potential, so the law remains a good approximation. I should probably just delete that out of this part of the lecture, but that's the idea. Okay, let's do the same thing for sodium now. Okay, so sodium, as we know, has a concentration that is much higher in the extracellular fluids than it is in the intracellular fluid. So we very clearly have a concentration gradient for sodium that is going to push sodium into the cell. As sodium comes into the cell, it will make the intracellular fluid more positive, and so that will create now an electrical gradient that would sort of push that sodium back out. So same as we had for potassium, we have these two opposing gradients, right, that are acting on sodium. At what point will these two gradients be balanced? Well, the answer again is at the equilibrium potential, right? So we have to work out what is the equilibrium potential for sodium. We use our Nernst equation to do that, right? All the constants and the temperature are the same. The charge for sodium is also +1, so we don't have to change that. The only thing that's changing, therefore, in this equation is the concentrations of sodium inside and outside the cell. And again, I just kind of eyeballed it from the figure, and then we do the math, and what value do we get? Positive 72 millivolts. So that's the equilibrium potential for sodium, right? That basically, with this given concentration gradient, 150 versus 10, in order for there to be a balance between that in order for there to be a balance between that concentration gradient and the electrical gradient, you would need a membrane potential of positive 72. It has to be 72 millivolts more positively charged inside the cell than outside. At that point, sodium is getting pushed out of the cell by that electrical gradient at the same rate that it's being drawn into the cell by this concentration gradient. And once again, as I was saying before, if we have a membrane potential that is not positive 72, in this case, then ultimately sodium is not at equilibrium, and it's going to move across the membrane in whatever direction is necessary to get it to equilibrium. So on Tuesday, for example, we saw that the typical resting potential for a neuron is, say, negative 70. Negative 70 clearly is not equal to positive 72. So what direction will sodium have to move across the membrane to get itself to equilibrium? You want to feel this one too? Has to come in. If it's negative 72 inside the cell, or negative 70, and sodium wants to get to positive 72, then sodium's got to come in, bring its positive charge with it, try to push things up to positive 72. Okay? We'll do this one more time, right? We're going to really kick this horse till it's gone. Here's chloride, which is another major ion in terms of resting potential. So again, we already know that chloride concentration is much more abundant in the extracellular environment than it is in the intracellular fluid. So we have a concentration gradient favoring chloride to come into the cell. As it comes in, obviously, it's going to make the interior of the cell more negative and the outside more positive. And so now we'll have this electrical gradient that will form that's going to try and push that chloride back out. And so we have, again, these two opposing gradients acting on chloride. And we can ask the same question we've been asking. At what point will these two gradients be equal? And the answer is the equilibrium potential. So we get out our trusty Nernst equation and our favorite calculator, and we do the math. Again, the constants in temperature aren't changing here, but the charge is different for chloride. It's -1, so I wanted to highlight that. And then again, we just put our concentrations in here for chloride in the extracellular and intracellular environment. We get out our favorite calculator, we punch in our numbers, and we get an equilibrium potential of -84. So again, that tells us that once the inside of the cell is 84 millivolts more negatively charged than the outside of the cell, that's the point at which the electrical gradient acting on chloride will be exactly balanced by the concentration gradient acting on chloride, and you'll have no further net movement of chloride across that membrane. Okay? So again, don't have to belabor the point too much, I don't think, at this point. If the membrane potential at a given time is not equal to chloride's equilibrium potential, then clearly chloride is going to be moving across that membrane trying to get to the equilibrium potential. All right? So what I hope you can kind of see here at this point is that it's by having these different concentrations of ions in the intracellular and extracellular environment, and the fact that these ions then want to move across the membrane down their concentration gradients, that's what's establishing the membrane potential. Right? That's what's causing charge separation to occur across the membrane, is the movement of these ions down their concentration gradient, and they will keep moving down their concentration gradient until they create a membrane potential that is exactly opposing their concentration gradient. And then they can't move any further because they're being pushed out and drawn in at the same rate. Okay? Is that okay? Are we clear with that? All good? Now, the next thing to point out here is, obviously based on the Nernst equation, if the concentration gradient changes, if we change the concentration of chloride inside or outside the cell, obviously we're going to get a new equilibrium potential. So if, for example, we were to double the chloride concentration outside the cell, push it from 115 up to 230, let's double it. Obviously, if you have a much bigger concentration gradient, you're going to need a much bigger electrical gradient to oppose it. So we would predict in that case that the equilibrium potential for chloride would become some bigger negative number. Right? We could do the math and figure it out if we wanted to. We won't, but it would become some bigger negative number. You would need a stronger electrical gradient to oppose a stronger concentration gradient. We also need to appreciate that temperature is part of this equation. And so changes in temperature, as could occur, say, in a fever, as could occur if you fell off a boat into the cold water and you were hypothermic, changes in temperature also change the equilibrium potential. And because temperature is in the numerator here, we're basically saying that as the temperature increases, the magnitude of the equilibrium potential will also increase. The sign won't change, but the magnitude of that number will. So for example, if we were to take the temperature from 310 Kelvin and let's say we push it up to 315 Kelvin, you got a bit of a fever, you'd expect the equilibrium potential for chloride to go to some bigger negative number. Maybe -96 or something like that, okay? Now, in terms of trying to explain why does temperature have this effect, what we have to keep in mind again is that we've said it's the movement of ions across the membrane that generates the resting potential. Well, temperature speeds up the movement of ions and particles in general. So the basic idea is that if you have a higher temperature, then because ions have more energy to move with, they're able to follow their electrical gradients with more like gusto, shall we say. And so then they're going to need a stronger electrical gradient to oppose them, right? It's kind of like if you imagine that, you had a rope tied around me, and at the beginning of the day when I've got lots of energy, I can really run fast and you got to pull hard to keep me back. By 7 o'clock at night, when I'm done my 4th lecture of the day, it'll probably be a much easier task to kind of pull me back, right? Because I'll have depleted my energy. So the idea is that at higher temperatures, right, because these ions have more kinetic energy to move, they're able to follow their concentration gradients more easily, and so you need a stronger electrical gradient to oppose them. Also, if you again, this is I think a very interesting point, if you imagine that we're at absolute zero, and so the T goes to 0, what's going to be the value for equilibrium potential? 0. And so that's a powerful reminder for us that if we have no movement of ions across the membrane, if we're at absolute 0 and the ions are motionless and can't cross the membrane, we won't have a membrane potential no matter what the concentration gradients are, right? Doesn't matter how much energy I have to run, if my feet are glued in place, you're not going to have to exert any energy to pull me back. So that's another important thing to kind of keep in mind here is the effect that temperature has on membrane potential. Now the next thing we have to kind of appreciate here, and I kind of guess was hinting at this a little bit earlier, is that ions, they can't just freely cross the phospholipid bilayer. Right? This is just your typical kind of first-year textbook figure reminding us that we have this hydrophobic core in our plasma membrane, and so charged species cannot just pass through the hydrophobic core. And so when we've been talking about all these ions moving across the membrane, creating this charge separation, how are they getting across the membrane? Well, the answer is they're getting across the membrane through what are called leak channels. They can't just cross through the plasma membrane. They can only cross through if there are channels to permit them to pass. And so at this point, I'm introducing you to what are known as the leak channels, right? So you can see here we have sodium leak channels that let sodium across the membrane. We have potassium leak channels that let potassium across the membrane. There are chloride leak channels that let chloride across the membrane, for example. So the ability of ions to move across the membrane depends on the presence of leak channels. One characteristic of these leak channels is that they're always open, right? So under all circumstances, they are open, so they're always allowing for this kind of baseline movement of ions across the membrane, which allows then for these ions to again, move across the membrane, following their concentration and electrical gradients, as we've discussed. Now, if you look at this particular figure, you might think to yourself, why did the person who illustrated this figure, not just have equal numbers of these different leak channels? There's probably somebody who's got a little bit of, kind of neuroticism about this, oh, there should be 1 sodium for every 1 potassium. It looks so nice and symmetrical. But the reality is that the reason the illustrator did what they did is because there aren't equal abundances of these different ion leak channels in the membrane. In fact, on a typical cell membrane, you'll find that for every 1 sodium leak channel, you have about 100 potassium channels and about 10 chloride channels. Meaning that our membranes are inherently way more permeable to potassium than they are to either chloride or sodium. And so now with that information, you're wondering, why didn't this illustrator draw 100 potassium channels to that 1 sodium channel? Right? Were they just fucking lazy and didn't want to have to draw out another 96 of these channels? Probably that was the case. Or maybe it was AI and it didn't understand what it was doing. Who knows? But in any case, we have to appreciate that, again, ions can only cross the membrane through these channels, and these channels are not present in equal abundance. So some ions can move to follow their concentration and electrical gradients more easily than can others. And so then that brings us to kind of the summation. And I'm going to run out of just enough time here, so I'll just give a quick little summation, and we'll probably pick this up tomorrow. But kind of where we're at this point is we've looked at these 3 ions, sodium and potassium and chloride. We've calculated for each what their equilibrium potential is from the Nernst equation, and we saw that they're very different. especially for sodium, right? It's at positive 72, and the others are much more at this negative 80 value. So what we can hopefully quickly realize here is, okay, well, these different ions, they all have slightly different equilibrium potentials they want to get to, right? So there can only be, at any given time, one resting membrane potential value. So if each of these three ions has different equilibrium potentials that it wants to get to, I mean, how do they figure out what the one value is going to be? Do they just like duke it out? Like little sodiums punching little potassiums, trying to figure out who's going to win? The answer is obviously not that, but we'll have to wait till tomorrow to find out what the answer is. So for now, you can at least go home and think about little sodiums and little potassiums engaged in little fights with each other, and then tomorrow I can burst your bubble about how that works. Well, I hope that was on the whole time.